This is podcast 18. So we talked about breakdown of carbohydrates using glucose as an example, and then other carbohydrates can enter in different parts of glycolysis. Now we're going to talk about breakdown of fatty acids. This is known as beta oxidation. It is the primary pathway for breakdown of fatty acids in most vertebrates. It happens in mitochondria and in peroxisomes. Peroxisomes are another organelle that has the enzymes for beta oxidation of fatty acids and also participates in detoxification of peroxide radicals. And beta oxidation requires oxygen. So you typically find high capacity for beta oxidation in tissues that have high energy demand, tissues that have lots of mitochondria in their cells, like liver tissue, heart tissue, oxidative muscle. These are all excellent at beta oxidation. This corny picture of people doing aerobics is to remind me to tell you that this is how aerobics got its name. So back in the 80s when aerobics became a fad, the idea was that you are working at low impact, relatively high reps, and within the ability of keeping your muscles supplied with oxygen. So you don't go past that ability like in a sprint or in a power lift, but you stay within that ability of supplying oxygen to your muscle. And in order to do that, you do that in order to burn fat, because these are people interested. Initially, aerobics was for people interested in weight loss, and you have to be able to supply oxygen to your tissues in order to break down fatty acids that are stored. So that's where aerobics gets its name. The primary form in which fats are stored in vertebrates is triacylglycerol. Most vertebrates, not all vertebrates. Triacylglycerol has a glycerol backbone and then 3 acyl chains, 3 fatty acid chains hanging off that backbone. And the enzyme that breaks that off is triacylglycerol lipase, or TAG lipase, that breaks off a fatty acid one at a time. If it breaks off one fatty acid, it becomes diacylglycerol, 2 acyl chains and glycerol, and then a free fatty acid, and then monoacylglycerol, and then finally just glycerol. This is mobilizing the fatty acids that is stored in your fat tissue. And the glycerol that is mobilized, you can also break down. And glycerol can enter glycolysis. Here's glycolysis, okay? Here is glucose. Here's glycogen breaking down individual glucose units, glucose 1-phosphate, glucose 6-phosphate, then fructose 6-phosphate, fructose 1,6-bisphosphate, and so on. You recognize this. Glycerol can enter into the part after fructose splits from a hexose, a 6-carbon sugar, into 2 3-carbon sugars. Glycerol can enter glycolysis into that pathway, and then you can utilize glycerol for energy, going all the way to pyruvate, and then generation of acetyl-CoA, TCA cycle, electron transport chain, etc. Okay, if you have mobilized a free fatty acid by the action of triacylglycerol, then that free fatty acid can't be directly imported into the mitochondria. So this is the mitochondria depiction of its outer membrane and its inner membrane and then the matrix. That free fatty acid has to be activated first by adding a CoA to it, and then it becomes a fatty acyl CoA, okay, acyl CoA. So something like palmitic acid, which is 16 carbons, no double bonds, that would get a CoA added to it, and it would become palmitoyl CoA, and then carnitine. That acyl-CoA becomes acyl-carnitine. Switch out the CoA for carnitine. That allows the fatty acid molecule to transverse the outer mitochondrial membrane, and then that acyl-carnitine will transverse the inner mitochondrial membrane through an acyl-carnitine translocase, and then finally the carnitine is taken off once it gets into the matrix, and it goes back to an acyl-CoA molecule. So again, fatty acids can't enter mitochondria on their own. They have to go through this conversion process, this activation process, where first they're converted into an acyl chain plus CoA. Then that is swapped, the CoA is swapped out for carnitine, transferred across the outer membrane, then the inner membrane, and then transferred back onto CoA, and now it's ready for beta oxidation. This is actually the rate limiting step, is getting it into the mitochondrion. There are two translocators that do that. One is commonly called carnitine palmitoyltransferase 1, CPT1, that's on the outside, and then there's CPT2 on the inside. Now, this is called carnitine palmitoyltransferase just because that most of this work was done on mammalian cells, and mammals have palmitic acid, 16 carbons long, no double bonds, as a typical substrate for beta oxidation. And so they said, okay, we're going to call this the thing that transfers palmitic acid across the membrane. Of course, it transfers all kinds of different fatty acids across the membrane, arachidonic acid, oleic acid, linoleic acid, etc. So a better name is carnitine acyl transferase. So you may see either term, CAT, or because it's just tradition, CPT. So even though it's called CPT, it doesn't mean it only transfers that one species of fatty acyl-CoA. It transfers all kinds of CoAs. But that's not to say there isn't a preference. So my PhD lab actually looked at this in some cold-bodied fishes and showed that they do have a preference for what kinds of fats they burn. They're not all equal for beta oxidation. Okay, so I'm going to talk about beta oxidation in a couple different slides. This is the slide, the picture from your book, which I think is kind of confusing. I'm going to show you a different picture after this one, and hopefully by looking at both of them, you'll get the idea. Okay, so beta oxidation is called beta oxidation because it operates on the beta carbon. So here's the carboxyl end of a fatty acid, and then here's the chain. Here's the first carbon group after the carboxyl end, and then the second one. So the first one is alpha, the second one is beta, and that beta one is key. So first you're going to activate the fatty acid by adding the CoA to get it transferred across. Okay, acyl-CoA dehydrogenase is going to do that. That's FA1, this enzyme right here, and that actually takes ATP. So that's important. And later on, I'm going to ask you about how many ATPs you get out of this process. You can calculate it based on the products coming out of this process, but you have to account for the ATP that it takes to activate it in order to get things started. Okay, so alpha carbon, beta carbon, okay, and then here's the carboxyl group. And enol-CoA hydratase here is going to add a CoA SH group to the end of this molecule, okay, alpha carbon, beta carbon. And then this link between the alpha and beta goes from a single bond to a double bond, okay, and then this beta carbon here gets a hydroxyl group added to it. So this is fatty acid molecule 3 or enzyme 3 in beta oxidation called beta hydroxyacyl CoA, fatty acid molecule 3. So this enzyme right here, 3, even though it's not rate limiting, sometimes you'll see this in a biochemistry paper as an indicator of how fast beta oxidation is proceeding, and that's just because the CPT activity is typically really, really low in cells. It can be so low that it's hard to measure, and so some people use this enzyme, FA3, beta hydroxyacyl CoA, instead because it has higher activity, even though it's not rate limiting. Okay, again, alpha carbon, beta carbon. Now we've got a hydroxyl group on that. That hydroxyl group gets changed out for a keto group by acyl-CoA acyltransferase here. And this acetyl-CoA, 2 carbons, okay, plus CoA, that comes off, and at the same time a new CoA SH comes in, gets added on to the end. So this right here, this bond breaks, and you get acetyl-CoA molecule, and that acetyl-CoA goes off to the TCA cycle. And then this a new CoA SH adds on to the end that you just lopped off right here, okay, right here, okay, and then you're done with one cycle of beta oxidation. So you're cutting off 2 carbons at a time, releasing them as acetyl-CoA, and then to do it again, you're going to cut off the next 2, and then in the third cycle, the next 2. In every cycle, you get FADH2, which is a reduced energy carrier that you're going to get the energy back from during electron transport, enters in complex 2, and then NADH here, which you're going to get the energy back from entering in complex 1 in electron transport. So if you have a fatty acid that is 16 carbons long, 22 carbons long, 18 carbons long, you're going to go through this cycle 8 times, 9 times, 11 times per fatty acid. And each time you go through this cycle, you get FADH2 and NADH. And so you can see that these are very energy-rich molecules, and you can get a lot of ATP out the other end. So again, the acetyl-CoA that gets released enters the citric acid cycle, just like we talked about before, when pyruvate is decarboxylated to make acetyl-CoA. Now those acetyl-CoAs are coming in from beta oxidation, and they can enter the citric acid cycle, and away you go through citric acid cycle, electron transport, and chemiosmotic synthesis of ATP through the FOF1 ATPase. Okay, so here is a H5P question for you that I'm not going to put any qualifications on, just read that straight. But the real answer is that it's less than the maximal yield of 18 ATPs per acetyl-CoA. And that's because at the beginning of that cycle, when you're adding the acetyl-CoA, you get, it takes ATP to do that. So that has to be subtracted out from your total yield. So now I'm going to look at another diagram about beta oxidation, and even though these aren't the same quality in terms of graphics, I think it is easier to see what's going on. So they break beta oxidation into these 3 stages. Stage 1 is the thing that we're just talking about, where here's the acyl chain here, and it's cutting off 2 carbons at a time. One here, another cycle here, another cycle here, another cycle here. Each one of those cycles enters the citric acid cycle. This one is 16 carbons long, so this would be palmitic acid. Each one of those enters the citric acid cycle. So you're going to go around this 8 times, and then you're going to generate lots of NADH, lots of FADH2, and then you're going to harvest the energy in those reduced molecules by taking them through the electron transport chain, creating a proton gradient, and making ATP. If we just look at stage 1, which is what the diagram in your book talks about, okay, here's the alpha carbon, beta carbon, and then adding on the CoA here, and then this R just means this goes on for 16 carbons. So a fatty acid beta oxidation enzyme 1, acyl-CoA dehydrogenase. What's going to happen is there's going to be a double bond here, and an FAD becomes reduced, FADH2, and then enyl-CoA hydratase here. You're going to add in water, and there's a modification of this group here, and you have a hydroxyl group added on here, and then beta hydroxy-CoA dehydrogenase is the one that I said scientists may use as an indicator of the ability for beta oxidation, even though it's not rate limiting, just because CPT activity can be really hard to measure. And you're going to charge up NAD to reduce NADH, okay? And you get this molecule here, and the hydroxyl group gets replaced with this keto group. And then finally, this acetyl-CoA comes off of this molecule, and a new CoA group adds on to where this came off. So bond is going to break right here. A new CoA comes on right there, and then this acetyl-CoA goes into the TCA cycle. And then you'd repeat this again, and now it would be, at the end of that, it would be 12 carbons long, and then 10, and then 8, and then 6, and then 4, and then 2, and then you're done. Okay? Okay. So, beta oxidation is super important, not just because of human obesity, but also because that there are organisms that rely on beta oxidation to make it through a long period of dormancy, like grizzly bears. So by the time you are watching this, you're going to be in Fat Bear Week 2020. It's one of my favorite things to see on the internet. This is from 2019, where this bear, Holly, was the winner of Fat Bear Week in 2019. So this is set up like March Madness bracket. People vote, and then week by week, they vote on who's going to be the biggest bear at the end of the contest. So this is the same bear. This is the same individual bear on July 12th, soon after emerging from hibernation, and then in September 22nd, gained an enormous amount of weight. And by enormous, I mean enormous. So a normal diet, according to FDA, is 2,000 kilocalories a day for an adult human. Bears can eat 20,000 kilocalories a day. So this is 10 times a normal human diet. They're not 10 times bigger than people are, although they are maybe 5 times bigger or 7 times bigger. But they can really, really eat. So when I say kilocalories, this is what you would see on a McDonald's menu or on a can of soda as calories. They just take the K off, but scientifically it's considered a kilocalorie. This can convert into gaining 4 pounds a day during this season. So what they're doing is they're really bulking up in preparation for hibernation. They want enough calories to make it through hibernation, where they're going to lower their body temperature, lower their metabolic rate, and just use stored fat and the energy in that stored fat and the process of beta oxidation to have them make it through the winter. So if they don't pack on enough, they're going to end up short and they will not emerge from hibernation. They'll die. So they have to go through this enormous weight gain in order to do that. And they're not the only animals that do that. There are all sorts of animals that go through this sort of percent change in body weight. Everything from birds to whales. You should watch the internet for Fat Bear Week 2020. It runs September 30th through October 6th. Here's another one sitting on the bench. They're super cute, super dangerous too. I've been closer than I wanted to be next to a grizzly in Alaska, and it really gets your heart racing. But there's a webcam set up on the Katmai Peninsula Conservancy website where you can watch bears feeding at the Katmai Peninsula on these salmon, and this is how they can just gain such huge amount of weight in such a short time. And we'll see who the winner is for 2020. Okay, so we know how cells break down carbohydrate as glycolysis as an example. Now we know how cells break down fatty acids with beta oxidation as an example. How do cells break down protein? Protein oxidation can also be a source of fuel to make ATP. We tend to think, because we're humans, we tend to think as protein as a last resort for breaking down stored fuel, but not all vertebrates think this way. Not all vertebrates evolved this way. But it is true that many will prioritize breaking down carbohydrate and fat when it's generating ATP. There are some tissues that have a preference for which fuel they want to burn. Like neural tissue, neurons are famous for preferring carbohydrate. But it's not that you go through all your carbohydrate and all your fat before you start attacking your protein. An animal that dies of starvation will still have detectable fat on the body. So it can't be that you go through all of this and then all of that and then protein is third. An example of an organism that catabolizes or breaks down their protein preferentially is a migrating salmon. So salmon will go through this Herculean migration from the ocean back to their spawning grounds, and during that time they will not eat, and they will and they're exercising the whole time. So they're swimming and not eating for as long as 11 months. And during that time, you can see that they're the enzymes that they're using to break down muscle, which is largely protein, the expression of those and the activity of those proteins that break down protein are increasing the further they get away, the further they get along during their migration. Here is another enzyme that is used to break down protein. You can see that increases the further they get away from, the further they get into their migration. LDH is an enzyme that decreases. So LDH is one of the enzymes that catalyzes what you do with pyruvate when there is not enough oxygen around. So we talked about that you can do a fermentation reaction in order to regenerate NAD. You use LDH to catalyze the conversion of pyruvate into lactate, and you regenerate free NAD so you can keep glycolysis going. So you're going to be doing less of that as your salmon moving through your migration. It's also an indicator of just how much muscle is there, and there's a breakdown of the muscle. LDH is typically high in muscle activity, and as the salmon is breaking down its muscle to use as fuel, there is less of this enzyme, which is an enzyme of muscle, in the evident in the salmon. Okay, so I'm not going to go over how all 20 amino acids are converted into ATP, but just to mention this generally, and this is what you're going to be responsible for, is generally how does it happen. So for these three amino acids, when those are liberated by the action of a protease or a peptidase, which is an enzyme that cuts the peptide bond in between amino acids in proteins. And if a free alanine is released, that can be converted into pyruvate, and then that pyruvate enters the stage 2 decarboxylation, generates acetyl-CoA, and then TCA cycle, and you would generate ATP, as you saw when we talked about glycolysis. Aspartate can be converted into oxaloacetate. Oxaloacetate is an intermediate in the TCA cycle. Glutamate can be converted to alpha-ketoglutarate. Alpha-ketoglutarate is an intermediate in the TCA cycle. So for these three amino acids, it's pretty simple to see how you can go from an amino acid to something that you can break down in the TCA cycle. For the other amino acids, it's a more complicated pathway, but the idea is similar in that you're going to convert it into something which is an intermediate of the TCA cycle, and then you're going to use the TCA cycle to completely oxidize it. And you're going to see this, hopefully you're seeing this already, is that the TCA cycle, also known as the Krebs cycle, also known as the citric acid cycle, is sort of like a metabolic crossroads in the cell, in that if you're breaking down carbohydrate, you're going to use that cycle. If you're going to break down fat, you're going to use that cycle. And if you're breaking down protein, you're going to use that cycle. Okay, H5P question for you. Okay, as you're studying, you should be able to trace the carbons going from stage 5, going through wherever you're starting, all the way through stage 5 of oxidative phosphorylation. Stage 5 would be going through the FO-F1 ATPase. Okay, so if you start with glucose, if you start with a fatty acid, if you start with an amino acid, you should be able to take that all the way to making ATP going through the FO-F1 ATPase. Okay. And I say 3 groups, everybody should talk. This is left over from when we were in person. But what I would try to do is try to do this in a study group. Try to do this yourself. Imagine you're going to get this on a test. Hint, hint.